The wrappings were so well preserved that the first researchers to examine them mistook the body for someone who had died within living memory, until the carbon dating came back and the number printed on the lab report stopped the room cold. Not decades, not centuries, 5,400 years. The linen was still pliable. The facial features were still distinct. The individual lying before them had walked the earth before the pyramids existed, before writing had fully taken hold, before any civilization we recognize by name had risen to dominance. And yet there he was, complete, coherent, legible in a way that ancient remains almost never are. The researchers documented everything: the posture, the burial goods, the isotopic signatures in the teeth. They assumed, as generations of researchers before them had assumed, that this person was an ancestor of someone alive today, that somewhere on Earth people carried forward what he had carried. They were wrong, not partially wrong, not wrong in nuance or in detail, wrong in the most complete and fundamental way science allows. The DNA this man carried, the precise molecular signature locked inside his preserved cells, matches no living human being on the planet, not in any database, not in any population, not even as background noise. His lineage ended, completely, and he is not alone. For more than a century after systematic mummy studies began in earnest, the story of who these ancient preserved individuals were seemed to follow a tidy logic. Physical archaeology built the framework, burial practices linked to known cultural traditions, tool technologies traced back through time to identifiable populations, pottery styles connected sites across continents. The scholarly consensus, reinforced through generations of careful excavation and comparative anatomy, held that the world's oldest mummies, particularly those found in the hyper-arid fringes of the Atacama Desert in South America, the Tarim Basin of western China, and the salt-preserved bog bodies of northern Europe, represented early branches of lineages that ultimately continued forward, ancestors, in other words, of modern populations. The Chinchorro mummies of northern Chile, the oldest artificially mummified human remains ever discovered, were assumed to be ancestral to Andean indigenous peoples. The Tarim Basin mummies of Xinjiang, with their startling physical preservation and unexpected features, were linked to Central Asian steppe populations whose descendants still exist across Eurasia. The Danish bog bodies were connected backward to Proto-Germanic populations, whose genetic legacy runs through millions of Northern Europeans today. It was a coherent picture, logical, supported by geography, by physical similarity, by the reasonable assumption that people who lived somewhere left descendants in that somewhere. The textbook version went like this. Ancient mummies are old members of ongoing stories, frozen chapters, not closed books. Radiocarbon data from El Morro, Camerones, and Arica consistently place the earliest Chinchorro mummies between 5,000 and 7,000 BCE, making them older than the Egyptian funerary tradition by nearly 2,000 years. Archaeologists from the Universidad de Tarapacá spent decades documenting the sophistication of Chinchorro mortuary practices, the removal and reconstruction of internal organs, the use of clay masks, the deliberate treatment of the dead as objects requiring preservation and care. These were not incidental remains. These were people whose communities invested extraordinary effort in keeping them present. The assumption of biological continuity with modern Andean populations felt not just reasonable, but almost necessary. Who else would they belong to? The same logic applied in Xinjiang, where physical anthropologist Victor Mayer of the University of Pennsylvania had spent years arguing for a Western Eurasian origin for the Tarim mummies, Bronze Age people with light hair and tall stature, preserved in one of the driest environments on Earth. The consensus was these were migrants, yes, but migrants whose genetics would show up in the mixed populations of Central Asia today. The story had ancestors and descendants. It had continuity. Then DNA arrived. In 2021, a team led by genomicist Chung Guanzhong at Seoul National University, working in collaboration with Fu Qiaomei at the Chinese Academy of Sciences, published a landmark study in Nature analyzing the genomes of 13 mummies from the Tarim Basin dating between 2,000 and 4,000 years old, some of the most intact ancient human specimens ever subjected to genomic analysis. The extraction process was extraordinarily difficult. Ancient DNA degrades in a pattern that looks, under analysis, like a document fed through a shredder and then left in a hot car for 1,000 years. The fragments are short. The damage is chemical and cumulative. The researchers required samples drawn from the petrous bone, the densest portion of the inner ear, where ancient genetic material survives longest, protected inside what amounts to a biological vault no larger than a sugar cube. From that vault, they pulled enough endogenous DNA to run full genomic sequencing. What came back defied the existing model so completely that the team cross-checked against 127 ancient Eurasian populations and over 200 modern reference genomes before committing to publication. The Tarim mummies showed zero admixture with any neighboring population, no steppe ancestry, no Iranian farmer ancestry, no Siberian hunter-gatherer ancestry, nothing from east or west or north, They were, in genomic terms, an island, a population that had existed in isolation for so long and then vanished so completely that their genetic signature had left no trace in any living person on Earth. The paper named them plainly an ancient North Eurasian population that had survived in genetic isolation since the Pleistocene, since the Ice Ages, since a time before most modern human populations existed in recognizable form. Not a migration, not an admixture, a remnant, ancient, sealed, and gone. This is where the data becomes genuinely staggering. The Terry mummies carried a haplogroup profile, a specific set of Y chromosome and mitochondrial markers. That represents a genetic lineage branching off from the main trunk of modern human variation, somewhere between 9,000 and 13,000 years ago, during or immediately after the last glacial maximum. That is not a typo. These individuals belong to a population that had been genetically distinct, and apparently reproductively isolated for somewhere between 9 and 13 millennia before the oldest of them was placed in the sand. Autosomal analysis, the examination of the full genome rather than just mitochondrial or Y chromosome markers, confirmed the finding with a clarity that ancient DNA studies rarely achieve. Comparing 13 ancient genomes against a reference panel of 327 modern and ancient populations produced no significant match. The technical term geneticists use is basal. It means foundational. It means the branch split off early and then developed in isolation, accumulating mutations that exist nowhere else. No modern Uyghur population carries this signature in meaningful proportion. No Kazakh, no Iranian, no Han Chinese, no Siberian population carries it. The lineage did not mix outward and dilute. It ended. The Chinchorro data tells a companion story with equal force. Ancient DNA recovered from Chinchorro mummies by Lars Fehren-Schmitz at the University of California, Santa Cruz, and published in Science Advances, revealed that the earliest individuals, the ones practicing the most elaborate mortuary rituals, the ones whose descendants presumably carried the tradition forward, show genomic profiles that diverge sharply from later Andean populations at the same sites. Not a gradual shift, a discontinuity. Something happened and the earliest people were replaced, not continued. Here is what that means in practical terms. A woman, perhaps 30 years old, perhaps younger, her age estimated from dental wear and bone density, was buried in the Atacama with her infant child, both their bodies prepared with the full ritual sophistication of Chinchorro mortuary practice, clay reinforcing the facial structure, ochre dusting the skin, grave goods placed with evident intention. Her community spent hours, possibly days, on her preparation. They did not do this for strangers. They did not do this for outsiders. This was one of their own, given every care their tradition prescribed. And then, somewhere in the millennia that followed, the people who remembered that tradition, who carried its biological inheritance, who might have said she was one of us, they vanished. Not from the archaeological record, which continued at the same sites with similar tools and similar burial orientations. From the genome, the descendants of the people who built the Chinchorro tradition were replaced by people who adopted the tradition but did not share the biology. The story persisted. The storytellers did not. In the Tarn Basin, the case is even starker. No cultural continuity survived to mask the genetic break. The Bronze Age individuals, with their perfectly preserved woolen textiles, their elaborate felt hats, their grave goods suggesting a fully developed and internally complex society, they left nothing forward. Not a craft tradition, not a linguistic trace, not a genetic whisper in the populations who later occupied the same land. They existed, elaborately and unmistakably, and then they were gone. No compass, no descendants, nothing but preserved bodies and salted earth and a genome that matches no one alive. The mechanisms of such complete disappearance are not mysterious to population geneticists, even if they are difficult to absorb emotionally. A population does not need to be annihilated to vanish genetically. It needs only to stop reproducing at replacement rate while neighboring populations expand. Epidemic disease can do it, and the archaeological record in both the Atacama and Central Asia shows signs of population stress in the relevant periods. Climate shift can do it. The Tarim Basin was already hyper-arid. Any sustained change in the water table feeding the oasis, systems would have been catastrophic for a small, isolated population. Absorption can do it. A genetically distinct group intermarrying into a larger population until their markers dilute below detectability across enough generations. What makes the Tarim mummies unusual is the completeness of the erasure. Most ghost populations leave traces. They show up as a few % of ancestry in regional populations, a signal detectable only through sophisticated admixture modeling. These people left nothing. The isolation that preserved them genetically, that kept them a distinct, coherent population for 13,000 years, also meant they had no reservoir of mixed descendants to carry pieces of them forward. When the population ended, the lineage ended with it. Meanwhile, the Chinchorro evidence points toward something darker, a population that was present, that was practicing complex cultural rituals, that was apparently thriving in a challenging environment, and was then replaced by genetically distinct people who took up their material culture while contributing none of their DNA to the region's biological heritage. The original people were not ancestors. They were occupants of a chapter that was closed and overwritten. This matters beyond the specific sites and the specific mummies. The pattern of complete genomic erasure, of populations that left physical evidence but no biological legacy, is turning out to be far more common in the ancient record than anyone suspected before large-scale ancient DNA studies became possible. David Reich at Harvard Medical School, whose laboratory has produced some of the most consequential ancient genomics papers of the past decade, has argued in multiple publications in Nature and Science that the model of gradual population continuity that dominated archaeology for most of the 20th century is simply wrong. The ancient world was not a slow accumulation of cultures passing knowledge forward across generations. It was a series of demographic disruptions, population replacements, complete extinctions of isolated lineages, mass migrations that overwhelmed existing genetic structures almost entirely. The Yamnaya expansion from the Pontic steppe around 3000 BCE, for instance, effectively replaced between 50 and 90% of the ancestry of Western European populations within a few centuries. The people who built Stonehenge are genetically unrecognizable in the modern British population. They were replaced. The people of pre-Neolithic Europe, the hunter-gatherers who occupied the continent for tens of thousands of years, persist today as only a small fraction of background ancestry in some populations, not because they were always few, but because they were overwhelmed. What the oldest mummies reveal is that this pattern extends back further than even Reich's model anticipated, into Pleistocene-era populations that had persisted in isolation for 10,000 years or more before vanishing without descendants. We carry, in the aggregate, the genetic inheritance of the populations that survived and expanded. Written into the cells of every living person is the accumulated legacy of people who reproduced successfully, who found ways to outlast their circumstances, who left children, and those children left children until the chain reached the present. But that chain excluded entire branches of humanity. People who were just as real, just as complex, just as biologically complete as any living person. People who made tools and wrapped their dead and navigated environments that would kill most modern humans in days, simply stopped contributing to the ongoing story. The genes you carry did not come from them. Nothing about your immune system, your metabolism, your height, or your sleep patterns connects to those people. They are not your ancestors. They are something stranger and more affecting than ancestors. They are our extinct contemporaries, people who existed alongside the populations that became us, who lived and died in the same broad era of human history and who left no one. The ancient DNA revolution has given us the ability to find them, not their stories, which are largely lost, but their biological signatures, their molecular presence, their proof of existence. And in finding them, we are confronted with the full and unsettling scope of what human history actually was, not a story of continuity, but a story of survival. Mostly, people died without descendants. Most human lineages ended. We see only the winners, and we have been mistaking the winners for everyone. The oldest mummy in the Chinchorro collection, a child no more than 2 years old, its small body prepared with the same ritual care as the adults around it, is currently housed in climate-controlled storage at the Universidad de Tarapacá in Arica, Chile. It has been there for decades. Its genome has been partially analyzed. The results are consistent with what the broader data shows, a lineage with no surviving thread. The child died more than 7,000 years ago in a community that cared enough to preserve it carefully, a community that presumably hoped, in whatever way communities hope without articulating hope, that the dead would persist. And they did persist, in a way that community could not have foreseen and would not have recognized as persistence, not in the living but in the preserved, not in descendants but in molecules, not in memory, but in sequenced genetic code, read by machines in the 21st century and matched against nothing. Somewhere in that same building, and in laboratories in Leipzig and Santa Cruz and Beijing, the physical remains of an erased humanity wait in drawers and freezers and archival boxes. Each one holds an archive. Each archive tells, in the language of base pairs and haplogroups, the same essential story. We were here, we were complete, we are gone. The ancient DNA field is still young. Fewer than 10,000 ancient human genomes have been fully sequenced out of the billions of individuals who lived in the deep human past. As that number grows, as new extraction techniques, as paleogenomics tools developed in laboratories led by researchers like Eske Willerslev at the University of Copenhagen and Johannes Krause at the Max Planck Institute, push the boundaries of what degraded samples can yield. We will find more of these erased lineages, more ghost populations, more severed branches, more people who left bodies but not descendants. The story of our species is not finished. It is only beginning to be read. And what it tells us, with every new mummy, every new genome, every new absence in the database of the living, is that the human past was larger, stranger, and more populated with lost people than we ever imagined. Subscribe to stay updated as we uncover them, because the drawers are full and the freezers are waiting, and we are only at the beginning of learning who we lost.